to talk today about one of my PhD projects, which is about the venom protein composition in true bugs and how it is influenced by the ecological niche. So let's talk about venomous insects. Insects are amazing. They make up more than 60% of all living animals. And they are very diverse, not only in their appearance, wait a second, but also in their biology and ecology. And venom use in insect has evolved at least 14 times independently. And I think the most famous venomous insects are probably wasps or bees. But today I want to talk about venomous true bugs. So what are true bugs? Most of you probably know these stink bugs, which are the typical true bugs. Probably don't have a very good reputation. But actually there are many more true bugs which can be very different also, or very diverse in their appearance and in their colors and also in their ecology. So these are all true bugs. They all look very nice, I think, and they're all pretty cool. And some of them, as I said, are venomous. So when we have a closer look at true bugs or Heteroptera, they belong to the Hemiptera, and then they can be differentiated into 6 different infraorders. And while all other hemipterans are exclusively phytophagous, Heteropterans have exploited a lot of different feeding styles. So you can see we have a lot of predatory true bugs, which are here in orange. Some have shifted back to plant feeding, like here or here, and we even have some secondary shifts back to predatory feeding styles. And we also have blood feeding Heteropterans. But not only their feeding styles are very diverse, also the habitats they have exploited. So we have a lot of aquatic or semi-aquatic Heteropterans, and also terrestrial Heteropterans, and you can see they are very diverse, and that's probably also why true bugs are so successful. So what all true bugs have in common are their piercing sucking mouth parts. So with this elongated rostrum, they inject saliva originating from the salivary glands into their food, and then they ingest the pre-digested and liquefied tissues through the rostrum into the gut. And in case of predatory true bugs, this saliva is venomous. So they have venom, and it can have paralytic, cytotoxic, and digestive activities. And with that, they use it to overwhelm, kill, and pre-digest animal prey. And they can also use the venom defensively, and that sting can be very painful. So it's very effective to deter predators. So when we have a closer look at the salivary or venom gland system, you can see that it consists of 3 parts: an anterior main gland or AMG, a posterior main gland or PMG, and an accessory gland. And today I will focus only on the AMG and PMG. So what we wanted to find out in this project is how the ecological niche influences the venom composition and activity in true bugs. And we mostly focused on habitat and feeding style, but today I will only focus on feeding style because of time. And we used different species for this project, and we focused only on the Nepomorpha this time, which are exclusively aquatic clade here in the Heteroptera, with predatory but also some phytophagous species. And we picked out 4 different species: Elaeocorus semicoides, Notonecta glauca, Nepacinerea, and Coryxa punctata. And although they all occur in the same habitat in ponds, they usually occupy different parts of this pond and different microhabitats. So while Elaeocorus, for example, and Coryxa are mostly on the pond bottom and between stones, for example, Notonecta glauca is usually on the open water surface, very exposed. It's also a very active predator, so it actively hunts its prey. And in contrast to that, Nepacinerea, for example, is usually very hidden in the vegetation. It's a typical ambush predator that just sits and waits for prey to pass by so that it can catch it. So you can see they're quite different in their ecology. And also their feeding style differs. So Eliocoris, Motonecta, and Nepa are very strict predators, but Coryxa belongs to the Coryxidae here, which have shifted to a mostly phytophagous feeding style. So from the literature, we found that Eliocoris semicoides probably feeds on small vertebrates, but mostly invertebrates, the same for Motonecta glauca. And Nepa cinerea, according to the literature, only feeds on invertebrates. And Coryxa punctata can also feed on invertebrates, but it feeds also on plants and detritus. So you can see also the feeding style differs a lot. So for this project, we used a proteotranscriptomic approach to find out how the ecological niche influences the venom composition and activity. So we used RNA from AMG, PMG, gut, and breath body, so from the two main glands and the gut and the rest body, to do transcriptomics, and then we did proteomics with AMG and PMG extracts and combined those data sets. We did differential gene expression analysis, interpro-scan, and signal P analysis, and with that we selected venom protein candidates, and then we could compare the venom protein composition of the different species, and we also conducted some activity assays. So now we first want to have a look at the general venom protein composition in those 4 aquatic true-bug species. So here I will show you stack plots of the venom proteins that we identified for each species here, for the AMG and for the PMG here. So it's absolute numbers of venom proteins that we identified with our approach. And they're grouped according to the putative function. And I don't want to go into detail here because then it gets quite complex. But just by generally looking at these plots, you can already see some differences. First, of course, in the number of proteins that we identified, especially here in the AMG, we have some with a lot of proteins and some with only 20 or 15 proteins, I don't know. And also when you just compare the general color patterns, you can also see that there's some compositional differences between the species, but also between AMG and PMG. And what you can also see is that a large part of the venom is this light blue color, which are enzymes that are involved in proteolysis, so proteases. And we want to take a closer look at the proteases. So here you can only see the proteases in the different species, and again for AMG and PMG, and they're grouped according to the protein family membership, according to the MIROPS nomenclature. What you can see is that the majority of venom proteins in most species belong to the peptidase family as one, which usually includes trypsines and chymotrypsines. And they are very common in many animal venoms, especially in venoms that are used for extraoral digestion, because you usually need a lot of proteases to digest protein-rich animals. But what you can also see that in Coryxa punctata, the plant-feeding bug, we find a lot of peptidase family C1 peptidases, which are cysteine peptidases, and those are usually common in phytophagous hemipterans like aphids or so. So you can already see that the phytophagous feeding style of this Coryxa punctata is reflected in the protease composition in the venom. And now we want to have a closer look at carbohydrates digestion. So carbohydrates, or here we only look at glycoside hydrolases, which are very important carbohydrates used for polysaccharide digestion. And they can be grouped into different families, which have different functions and activities. And again, only by looking at the general pattern, you can see Coryxa punctata has more carbohydrates or glycoside hydrolases than the predatory species, and also different families. So, as I said, carbohydrates are important for polysaccharide digestion, which means that they are also crucial for phytophagous insects in general, because plants contain a lot of polysaccharides which need to be digested. So it makes sense that, again, this phytophagous species has more carbohydrates than the other species, the predatory ones. But there's one family, for example, the GH13, which is quite important for phytophagous species, but we also find it in the predatory species, except for Notonecta glauca. So these amylases usually are used to predict a phytophagous lifestyle in true bugs because they are so important to digest starch, which is the main polysaccharide in plants. So we wanted to know if they might also be beneficial for predatory species. So if we look at the major polysaccharides, storage polysaccharides in plants and animals, which is glycogen in animals and starch in plants, we can see that they actually look quite similar. So the branching is a bit different here, as you can see in the structure, but the basal sugars are actually the same. So we were wondering, maybe amylases can digest both glycogen and starch and therefore are beneficial for both feeding styles. So we conducted an activity assay to measure the degradation of both substrates. And indeed, we could show that that two of the predatory species here, Iliochorus and Nepa, and also the phytophagous species here, Coryxa, can digest both glycogen here in yellow and starch here in green. And Notonecta, the venom extracts could not digest any of the substrates, but we also didn't find any amylases in the venom, so that also makes sense. So what we could show with this assay is that salivary amylases in these true bugs can digest both glycogen and starch, the major polysaccharides in animals and in plants, and therefore they're probably beneficial for both feeding styles. And this is probably the reason why the predatory heteropterans did not lose this enzyme in their venom, but kept it because it can be beneficial and helpful. To summarize this, so we had a look especially on the feeding style, but also habitat and microhabitat, and wanted to find out whether this ecological niche can influence the venom composition. And indeed, it did influence the venom composition, but also the toxicity and also the digestion and digestive enzyme composition in true bugs. So we could only show this now for for aquatic true bugs, but probably this is also true for terrestrial true bugs. But we aim to find out more about this soon. So in general, we can say the ecological niche influences the venom composition and activity in aquatic true bugs. And with that, I would like to thank my team, especially my supervisors Heiko and Andreas, also my master student Sol, and my colleagues Natalie and Roy, which helped me with some experiments, and in general the Department of Insect Symbiosis at the Max Planck Institute for Chemical Ecology and the INPRS for funding. And thank you for your attention. And now I'm happy to answer any questions.
Thank you very much, Maike. Are there any questions from the audience? As we wait for some, I have some questions. What is the volume of the venoms you can get from one single bug? Or how many bugs you have to kill to get all the venoms? Or have they been killed?
Yeah, I mean, I can also collect venom from them without killing them, but for this for these experiments we used mostly venom extracts, so we had to kill them for that. Mm-hmm. And I can't really say which volume, because I usually just put glands in PBS buffer and then I have a dilution of it. But I would say depending on the size, there should be several microliters in there. Especially in the PMG. The PMG is usually larger than the AMG. From the AMG, we don't get so much. And also, I mean, in Coryxa punctata, the venom glands are a bit reduced, and we don't get so much venom out of it. Yeah.
Is there a reason why you didn't look at the accessory glands, or are they not responsible for the digestion part?
So the accessory glands are usually very small and they don't contain much protein and also not they don't contain much RNA, so probably there's not much venom that is produced there. And also in some other studies where they looked at which venoms are used for prey killing or for injection into prey and also for defense, the AG was never involved, so we did not focus on this one. Because then we would have needed to kill more insects to get enough RNA and protein from the AG. And we also looked at the venom, or which types of venom are injected into prey and which types of venom are used for defense. And that was mostly PNG venom.
Thank you very much. Are there any questions now from the audience? Then I have a last one. Can it also be necessary for some of the predators to inject amylase because the prey they hunt for is full of starch, so that they have to digest their food also before they can be eaten up?
That's in general also possible, but usually the prey that might be phytophagous and therefore ingests plants, and they have their own amylases in the gut usually. I mean, usually all the starch digestion takes place in the gut. So I wouldn't expect them to have their own amylases to digest the gut content of the prey. But of course it would be possible maybe, but I don't know if that's enough to really keep this amylase in the venom. I think it's more probably for the glycogen digestion.